Can you hear me better?
Yes.
Okay. So you'll soon be meeting Ananya Malcha, one of our research interns, in a moment, and also one of our patients who will be speaking today, Mark Wilhelmi. He's connecting by FaceTime, so we're putting all the pieces together. While this is happening, again, my name is Henry Colburn Stevenson Perez. It's hard for me to understand a lot of Chinese names, and I hope you feel the same about mine. So everybody just calls me Steve. Everybody calls me Steve. My patients, everybody. Please feel free to see me that way. It's an honor to be here today. I am also truly amazed at how quickly you organize, how quickly you come together. And I'm also impressed with how young you are. And I have a feeling that the ideas that you're having now will shape medicine for a long time to come. So please accept my sincere appreciation for the opportunity to chat with you. Are we ready to go?
Yeah.
Great. You're going to hear a presentation in a very different way than you've ever heard it before. You see my name here. Ananya Malcha will be speaking in just a moment. She's one of our research interns. And you'll be meeting Mark Wilhelmi in just a moment. Thank you. Thank you. I just learned a few days ago that Joe Goodman, who is a brilliant ENT cancer surgeon, is actually in another presentation right at this moment. He got commanded into this, and I'll be presenting his material. We have a lot of collaborators here, and this is one important idea. Please don't worry that you're going to see too much today. Don't worry. Lin Quan has graciously agreed to post this PowerPoint on the website and to send it to you. So you'll get everything. And you can play it back. You don't need to worry about taking notes too fast. In addition, as you're going to hear, we have a lot of references for you. Actual on-the-ground references that you can use for whatever your plans are in cancer, whether it's for clinician, researcher, patient, maybe some of you are caregivers, maybe some of you are in investment, market investment. You'll have all the references you need to master the field here. And those references are actually provided as links at the back of this PowerPoint in PDF form. So you can just take on the train and see it all, okay? It's really important for me to emphasize two things. I've been giving presentations for a long time. I've been with Walter Reed and the National Institutes of Health since 1974. And yet the presentation that you're going to hear today is very different than the one that you normally hear. What you're going to experience today is going to be more similar as if you were to come to see me as a patient. I'm an immunotherapy doctor at Walter Reed Hospital. Or if you were going to come to one of my lab meetings. It'll be that intimate, quiet, easy to understand. And at the end of the day, this day, the goal is to help you to be so powerful in cancer. That's what this entire seminar series has been developed to do. And by the way, it was all initiated by our youngest researchers. To the best of my knowledge, none of the people you're going to talk to have any conflict of interest to declare. And with that, we're going to start our presentation with some tools, some purely scientific tools that are designed to help equalize everyone, to make everyone equal. And these tools are ways to think about cancer that you've probably never ever been told about before. They were shocked when I heard about them. This is our first tool. The icon for it is here. And this is one of my favorite teachers. Can I see a show of hands of people who recognize this man? Can I see a show of hands? Okay. His name is Jonas Salk. Jonas Salk is, without a doubt, there's no debate about this, the greatest immunotherapist of all time. Number two, can I see a show of hands of what Jonas Salk discovered? Who knows? What was his biggest contribution? We have one hand. Yes, ma'am. The polio vaccine. That certainly was fun. But his greatest contribution was he was the man who prevented the flu pandemic of 1946. And you say, well, Steve, I didn't know there was a flu pandemic of 1946. I said, yeah, he prevented it. Are you aware of the flu pandemic of 1919? The infrastructure of the Western world declines, and boom, this horrible flu comes. World War II was much more devastating, and the scientists were certain that it would happen again. This was the man who developed the flu vaccine that you're receiving today. This is the man who has surely saved many, the pandemic of 1919 killed 20 million people. Hundreds of millions of people owe their lives to him. Would you like to hear what his message is for you today? Again, he's one of my favorite teachers, and this is what he says in the beginning. Are you ready? We're going to try to see if you can hear his voice here. We haven't actually tried this experiment. Ananya, you're going to try to play? We're going to be talking, in a moment, we're going to be doing an exercise called check-in. It's going to take about 5 minutes for you to sort of figure out what you know about cancer right now. And at the end of our second hour, we'll be repeating the exercise one more time so you can see how much you've grown in your knowledge of cancer. We've already gone through one of these tools, the 5, the power of scientific questions. And Ananya, in a few moments, is going to be talking about how 21st century scientists see science. Number 3, what is the biological basis of intelligence? The next one is, what do we scientists mean when they talk about learning? How would you measure learning? And finally, what is the new definition of health in the 21st century? These 5 key ideas are going to be central to the things that we'll be discovering together in this next 2 hours. We're going to be looking at a cancer immunotherapy case, the type of cases that Ananya and I and our team see every day. And we're going to analyze what happened from 3 perspectives: a pathology perspective, a surgical oncology perspective, and a patient's perspective. And then we go to break. And after break, when we come back for the second hour, we're going to be talking about how 3 questions that Ananya and her colleagues came up with a year and a half ago have exploded into 15 questions that we're offering to you. Questions you've never heard of before about cancer that might revolutionize the way you think as a patient, as a clinician, as a researcher, as a caregiver, or as a business investor. We're going to do checkout, and then we're going to do conclusions. And with that, if it's okay with you, we're going to jump into check-in. Lincoln, would you be able to, oh, you're already on top of it. We would like you to do about 5 minutes of hard writing. Is there anybody who doesn't have a pen? Is there anybody who needs a pen? Okay, Lincoln.
Yes.
Can I please?
Lincoln does have pen.
So Ananya, could you go on the other side,
Yes,
please?
definitely.
And Ananya has pens as well. Excellent.
We do encourage everybody to participate.
The purpose of this, while they're passing this out as quickly as possible, is for you to figure out in your own mind, what do you know about cancer? I'm going to put the questions down here now as the paper is coming out. The first question is, what is your definition of cancer? How do you define cancer? Number 2, what do you think the causes of cancer are? Number 3, what happens to a previously normal cell to cause it to behave in a cancerous fashion? Number 4, are cancer cells intelligent? Number 5, how can we prevent cancer? And this is the most important one, question 6. What is the most important question for cancer researchers to answer this year? What do you think we need to do? If you please take your paper and just fold it on the stars, we just want you to work with check-in at the very beginning, please. Just work with the front, just work with the top. And the only other key thing to get started with is to put your years of education right here. Do you see in this section right here? Years of education. If you have finished high school, that's 12 years. If you finished university, 4 more. That's 16. If you finished medical school, 20. Graduate school, postgrad training, put as many years as training that you have. And then you can jump in and start answering the question. Does everybody have a pencil? Does everybody have a paper? Okay? Ananya, is it okay if we get... Are we ready to get started? Do you need more papers over there, Linquan? Yes. Okay. Ananya, they need more papers, please.
Okay.
Okay, here we go. Everybody's getting a paper. Who needs papers, please? Okay. Why don't we get started right now? 5 minutes. Please write down as much of what you know about these 5 questions.
And it would be great if you could please participate in this conversation and in this dialogue today as one of the 300 million people who, number 1, have cancer. Do we have any cancer patients here today? Wow. Thank you so much for coming, and thank you so much for raising your hand. I'm sure it's very, very stressful, and I'm sure that everybody here is hoping for you to regain your health back. The second group of people that we really want to reach out to are people who have loved ones with cancer, including family, close friends, colleagues. Does anybody have loved ones with cancer? Wow, that's quite a few people. Thanks so much for raising your hand again. And I'm sure this is very, very stressful. And during this course of 18 months, I've come to learn that an individual doesn't get cancer, a whole family gets cancer. So I hope that your loved one gains his or her health back very soon. The third group of people that we want to reach out to, people who simply don't want to get cancer. Who doesn't want to get cancer? Wow, that sums up pretty much everybody. So I guess we're all in this, right? Okay, so our empowerment tool number 1 is, what exactly is the science of science? I know this sounds very easy and almost astonishing at the same time. All scientists do is measure the motion of matter. Scientists are looking for motion patterns that reveal entropy, which is everything moving in maximum randomness and lowest energy. And at this exact same time, scientists are also looking for motion patterns that predict sustainable health and meaning, which is the opposite of entropy. Component parts coming together to make the whole structure more complex and more sustainable. Science always has a truth-seeking focus, and all the scientific discoveries that survive falsification testing always become reduced to math equations. And you can think of that. How odd is that? That always happens. And every truth that scientists can reduce to mathematical equations meaning that it has no value, becomes the next level of truth for everybody. And other scientists also keep testing it all the time, which is the process of reproducibility. Our third scientific research authority is how do scientists describe intelligence? Scientists always start with an assumption. The assumption that we're starting with here is we live in one interconnected, interdependent universe. And this interconnected, interdependent universe is always sponsoring the forces of entropy. Everything moving, again, at maximum randomness and lowest energy. At the exact same time, our one interconnected, interdependent universe is also sponsoring the forces of sustainable complexity and health and meaning that has made our universe more complex in fits and starts. Do you have a question here? Okay.
By the way, if you have questions, please feel free to interrupt. Just raise your hand. What I'll be doing and what Ananya is going to be doing, if we can answer the question quickly, or if someone else in the audience can answer the question quickly, we'll answer it. But more likely, we'll record it. The way we went from 3 questions to 15 was by getting the feedback from the audience. That's how it all happened. So if you have a question, raise your hand and we want to make it interactive.
At the same time, while sponsoring entropy and while sponsoring health and meaning, that makes our universe more complex. Our one interconnected, interdependent universe is constantly sponsoring the forces of chaos. Usually people tend to confuse chaos with being entropy, but that's not true. Chaos can be seen as a combination of entropy and health and meaning. Now, I don't know if you guys have heard about these NASA scientists who are looking for intelligent life on another planet, and they're called Earth 2.0 scientists, so I'll be referring to them throughout this conversation. The way they describe intelligence is every organism that is able to predict the entropic forces that are always present in the chaos and the health and meaning forces that are always present in the chaos, and start moving towards the health and meaning forces. That motion is intelligence. Intelligence is just emotion. I want to emphasize that. Here's a perfect example that these NASA Earth 2.0 scientists would classify as intelligent. Remember, they're looking for intelligent life on other planets. So if you look at this picture at A here, this is a picture of a resting slime mold. This is, of course, not a single-cell organism the way it's displayed here. There's several single-cell organisms here. This is its resting position in a maze. If you look at B, when scientists decided to put 2 food sources on 2 different ends of the maze, the slime mold are able to find their way to the food sources. If you look at C here, the slime mold are starting to recognize what is the efficient way to get to the food, and they're starting to eliminate the blind alleys that don't lead to food. They're becoming more efficient. And D, they've completely let go of the blind alleys that don't work. So what is this but intelligence? And this is, again, something that's described as intelligent organisms through the NASA scientists.
And most impressively, if you take a brand new slime mold or amoeba and drop it into this, they've never seen the game at all. They will follow the lead of the people. They know how to remember and teach one another. They won't find any blind alleys. They'll learn together.
This is another example of intelligence inside of your body right now that's working perfectly for you. This is a white cell chasing a bacteria, and this video is actually speeded up all the time.
Gotcha.
So hopefully you can see that there are no communication systems that are working in this cell. It's only making motions that are moving away from entropy and are moving towards health and meaning. Because if it was making all those cell communications that you keep hearing about, then
You mean
the
down to
bacteria
DNA?
would get away.
You mean down to DNA?
Yes,
This
it
is-
has to go from the cell membrane down all the way down to the DNA, and that process just takes too long. By that time, the bacteria would get away. Our scientific empowerment tool number 4 is how do scientists define learning? Here I have a tool for you. I didn't want to be as dramatic at the same time. This really works for me.
You're doing good.
Here are several surge projectors, and this is a model that works really well for me in understanding how scientists really describe learning and how scientists can measure learning really, really quickly. So neuroscientists define learning as active unplugging and active replugging.
She's holding your mic for you.
Okay, thank you so much. Active unplugging and active replugging. This is all learning is. This is going on in your head right now. Neurons are detaching, unplugging, and reattaching with one another. And this process of active unplugging and replugging is called neuroplasticity. Thank you so much. I'm just going to put these to the side. Now that was at an animal level. What about on the single cell level? Well, just like the bacteria that you saw today, bacteria have an immune system called CRISPR. CRISPR knows how to work on 2 different levels. I know there's several experts in the audience who know much more about CRISPR than I do. At the same time, this is my basic understanding of it. As soon as a virus enters, on the first level, as soon as a virus enters a bacteria that has entered the bacteria before, then CRISPR just clips it and detaches it, thus killing the virus. So this is, again, unplugging and replugging. The second level that CRISPR knows how to work at is if there is a new viral infection coming into the bacteria, and if the bacteria survives that infection, then the CRISPR is going to make a nucleotide recognition code copy of that specific DNA from the virus, and it's going to store that copy in its own bacteria, in its own DNA, to keep a memory of that specific virus. So when that virus attacks, the bacteria can survive that. And these changes are actually passed on from one bacteria to another. For humans, most learning occurs at the epigenetics level. Again, I know that there are several experts in the audience who know about much more about epigenetics than I do. Again, this is my basic understanding of it. And please correct me if I'm saying anything wrong. So epigenetics is not altering the ATGs and C code in the gene. It's only suppressing and repressing the expression of those ATGs and C code. And it does that by processes called gene silencing using histones or methylation reactions. Histones get placed on the ATGs and Cs, and it also uses what are those called, Steve?
It's basic. All I know is basically histone. They use histones and methylation processes to silence those genes that are not helping the cell to do what it wants to do phenotypically.
Okay, and thank you so much. And these epigenetic changes, these silenced genes, can actually be passed on to through reproduction. So if you silence a gene for, let's say, diabetes, because you decide that that's going to go away now, you're not going to be diabetic, or you're not going to be prone to be diabetic like your parents are, then you can silence those specific genes using histones, and you can actually pass those on to your kids. And over time, those histones will actually alter the ATGs and C code, so you won't have the gene for diabetes at all. What 21st century scientists want to understand is how do cancer-fighting immune system cells learn, and we'll be exploring that a lot as we move forward in our conversation today. Our fifth tool is what is health in the 21st century? For humans, health is described as a biological process or motions that actually drive the overall health and meaning for that organism. Some health-promoting motions or genes are actually inherited. At the same time, organisms are also able to improve their health through epigenetics, which we just talked about. Evolutionary scientists measure health using the survival of the fittest tests. For humans, health has become progressively more complex and more scientific. In hunter-gatherer economies, they describe health in just one sentence. If you ask them what is health, how healthy are you, they're going to give you the amount of people in their tribe and the amount of animals in that specific environment and how healthy all of them are, just in one sentence. In agricultural economies, health is defined as, is this mic working?
Yeah, it's fine. You're doing fine.
Okay. In agricultural economies, health is defined as how functional are the humans in growing surplus and storing surplus. In Western industrial economies, we've developed this model of allopathic medicine, which basically is find disease and treat disease. At the same time, what if in 21st century scientists are finally starting to understand that it's a scientific impossibility to not have any diseases at all? It's a scientific impossibility to recognize all diseases in a specific organism and get rid of all of them. So our working definition for health today is enjoy living to one's full potential at any age while also enjoying enriching the quality of an individual's social network. I know we covered a lot, and we got through it, finally, and now we're getting into 2 minutes of comparing notes of what you guys have learned in these past 10 to 15 minutes about questions, the power of asking the right questions, because the answers pre-exist. The second one is the science of science, that all scientists do is measure the motion of matter. The third one is intelligence. It's simply a motion that drives constant chaos towards health and meaning, and not towards entropy. And learning is just active unplugging and replugging. Health is living to your full potential at any age while enriching the quality of your social networks. Now maybe what I said is true or maybe it's not, but now you get to decide that if this is really going to work for you or not. And with that, I think we can take a couple of minutes here to compare notes with one another, and then we'll be open for questions.
If we could take 2 minutes and talk to somebody next to you, just talk. You guys know a lot of stuff, okay? And now you see these tools. See what you think about them, and then after 2 minutes, we want to get your feedback for a couple of minutes and start the dialogue going back and forth. Ready? Go. Can you talk to your friends?
My name is Dylan. First of all, it's my personal opinion. I think it's a great discussion. And I think so. I have a few questions. The first of all, you said called NASA life scientist gave the definition intelligence of life. I disagree because those guys, maybe biologists, not health scientists. So there's other people like the National Institute of Health, they probably can give better definition for whether life or intelligence. So I think that's a crazy definition.
What would be a better definition of health? How would you define it better?
My definition is,
Excuse me, intelligence. The word is intelligence. What is the better definition of intelligence scientifically?
no, I don't know how to define the intelligence. The intelligence word very weak to life scientists, very weak. I mean, very complex beyond everybody's mind except God. I think that's my definition. the second question is in health. I think how define health. I think my understanding is understanding yourself, understanding your surroundings. That's the health.
That sounds like intelligence to me.
No, intelligence word really defined come from biophysicists, mathematicians, or engineers. That's, I think, it's poverty bias.
Okay.
Thank you.
Thank you. We have time for one more feedback. Who wants to jump in? That was wonderful. Who has something else? Yes.
Okay. It actually plays off that question of what is intelligence, because my question was, I'm sure people thought about, in terms of trying to look for extraterrestrial life, things like that, in very different ways than I have. The way I think of it is like through a nervous system. But I was wondering if maybe you couldn't broaden the definition of it and differentiate this evolution that occurs, let's say, at the DNA level, and call intelligence as maybe something that adapts at an epigenetic level.
Do you think that intelligence is uniquely human, or do you think it applies broadly?
Oh, it must apply broadly, but I mean, the question is how do you define it? Really?
Okay, and that's what this is exactly what we're hoping you'll be interested. You're going to be hearing these words used a lot. At least you know what we mean when we say it, okay? Start to see if there's something meaningful for you. We really want to end this period with a patient by the name of Mark Wilhelmi. So I want to move fast forward, if that's okay. Why should you care about any of this? Well, what if this was you? Or what if this was your father? This is the patient that we saw last year. This is his face. Point to the square. Yes, thank you. Thank you. This is his face and his teeth and his nose, and a giant melanoma attacking his skull and going down into his neck. And this was him 6 months later, all of this gone. His name is Bob McCulley. He has provided all of the HIPAA clearance to use his name. Everything's fine. He's a person worth remembering, just a remarkable human being. He's almost 90 years old. So I want to tell you briefly about this case. 89-year-old male, signed all the HIPAA documents, it's all clear. And he's a retired military pathologist, and he noticed a small patch of dark coloration for about 5 years, and suddenly, in 2015, it exploded with a growth in front of his ear that suddenly had swallowed up everything. By October of 2015, we had this fungating tumor that's called BRAF positive, and hopefully we'll have time to get into that. I'm not sure, but we hopefully will. And it was not resectable by that time. Next. And the patient refused any radiation therapy and any chemotherapy, including vemurafenib against BRAF. So we thought that anti-CTLA-4, so this is another one we want to get you in the language to work with, that this was going to be too toxic, and instead we started with anti-PD-1, which we'll talk about in a second. By 6 weeks, the anti-PD-1 had produced autoimmune disease against his thyroid, which was successfully treated. And by 10 weeks, what the actual melanoma lesions seemed to be growing. And then we started with Dr. Goodman a series of surgical biopsies. By week 12, the melanoma lesion began to shrink very rapidly. And by week 14, he had no evidence of disease. And this is something we're seeing with increasing frequency. Next slide. Okay, so one of the things that we will be trying to get across is this idea that we're at a tipping point in cancer now, where there's two different ways to think about cancer. This is the traditional way, version 1, cancer as a random terrorist event. As has been said here earlier, it seems to come from one cell. Next slide. And it seems to be because of bad luck. It was bad luck that random DNA insults caused proto-oncogenes, which we've heard about today, to become mutated oncogenes or driver genes like BRAF. Number 2, it could come from random mutations that silence tumor suppressor genes, as we heard today, like VHL or RB. It was also bad luck when the immune system randomly failed to identify and eliminate this cancer. And finally, if the cancer responds to treatment, it's also bad luck when it becomes, or good luck for the tumor, bad luck for the patient, when the random hypermutation causes the cell to become resistant. Let's talk about version 2. The team of scientists that I'm part of are wondering how much of cancer happens when immune system cells, that cancer is probably happening to everybody all the time. That I believe I have melanoma right now. And the reason I'm not a patient, I think, is because number 1, I have first responder cells, like mononuclear phagocytes here, that are patrolling. These are non-clonal amoeba-like cells. And then there's second responder cells, like B lymphocytes and T lymphocytes. And let's go to the next, we'll just pass B lymphocytes for now. We've been hearing about T lymphocyte clones for each non-self invader. And they, as you know, they go to the site and travel. And the clone that we're most interested in is the CD8 positive cells. For anti-CTLA-4, the mechanism for that is that when the mononuclear phagocyte antigen presenting cell presents antigen in concert with the histocompatibility antigens to the T cell, it also creates a binding here for, with CD8, CD28, and B7. These are co-stimulatory molecules. And at the same time, the activated T cell places a brake on itself, CTLA-4, that actually competes with B7, and when it has enough of it, it actually silences the cell. What we have developed is an anti-CTLA-4 antibody that covers this up so that it cannot bind to B7, and this is what causes unrestricted killer T cell activity. Unfortunately, it also produces autoimmune side effects. Next slide. This is anti-PD-1. Anti-PD-1, PD-1 is a molecule that's expressed on activated T cells progressively as it ages. And any cell that has a PD-L1 receptor, which could be a tumor cell, but it for sure is also mononuclear phagocytes, will eliminate it. What we do is create an antibody, anti-PD-1, that covers this up so that any cell that has a PD-L1 receptor cannot bind to it and eliminate it. It keeps this cell alive and fighting. This is what Bob McCulley received. The only problem is that the risk of producing autoimmune side effects is still very real. Next slide. What are the scientific factors that are associated with cancer? Well, we've talked about inherited unstable proto-oncogenes or tumor suppressor genes. Number 2, exposure to carcinogens, such things that damage DNA, like smoking or radiation or sunlight. Next one. Certain viruses, such as HPV and hepatitis B and C. And then obesity, but we don't know why obesity causes cancer. And the big question is, for us, for the second team, do these factors make cancer cells smarter, or do they make immune system cells dumber? And that's what the big debate is now coming to be. Next slide. let me show you briefly what happens inside of a tumor like this when you start to give PD-1. Number one, this is what you see when you biopsy a tumor like this. These are highly abnormal melanoma cells. If you see all of these mitoses, you can tell for sure that this is a cell that's dividing very, very fast, and this is the baseline tumor that the Macaulay tumor looked like. Next slide. When you look at a bigger section, this is what you saw, but this is what happens when PD-1 activates the immune system to start to clear it out. And this is a space where the battle is actually taking place. And we're going to drill down on this right now. What you see when you drill down is something amazing. Here's some intact tumor still, and here is tumor cells that are being broken apart. Here's a tumor cell that's undergoing apoptosis right here, is being surrounded by white cells. Here's the funny thing about these white cells. These are not T lymphocytes. These are first responder cells. Next slide. This is more of the same process going forward. And here's the surprise. When you stain these specimens for anti-CD8, you find there's very few CD8 positive cells. All these other white cells are first responders. This is the final biopsy in this case, where now all the tumor cells are gone. All you see is remaining white cells and fibroblasts, and you see the rebuilding of normal blood vessels to the tissue. Next slide. I am unfortunately going to go through, we're going to pass this interaction with Joe Goodman. He's the most amazing guy. He's the one that did these surgical biopsies on this case. And we're going to instead jump. Mark Wilhelmi, can he hear us? I don't know how much you've heard, but I know you've been a part of this presentation many times. If you could please take about 10 minutes. You're the last speaker in this session. Could you please tell a little bit about what you're learning about cancer from your perspective as a patient? And I want to emphasize to the audience that we consider cancer patients to be true experts, equals with the clinicians. And maybe you can tell a little bit about your journey.
Okay. First of all, I prepared a PowerPoint bullet presentation. I don't know if you can see it or not.
We can.
This is what I'd like to get across to the audience. Doctors are not gods. They're more like your personal trainer. Another bullet, ideally a team of healthcare providers should be available to each cancer patient located in close proximity to each other. You are your immune system. The immune system is unique to each person and should be nurtured and understood. Pathologists need to become more involved and provide more insight to the doctor and patient about the type of cancer the patient is fighting and an analysis of the patient's immune system. Cancer patients should take charge of the recovery in partnership with their health provider. Know yourself and your unique capability. My journey began, well, first of all, I'm a retired naval officer. I spent most of my career in the explosive force disposal community as an EOD officer. I've been out in the sun a lot, but I'm used to working with a team. We recruit from different specialties in the Navy, especially technical specialties like electronic technicians and ordnance people and radio operators and that sort of thing. We form our teams with an officer in charge or senior enlisted officer available and to dispose of bombs. We get together, we discuss how we're going to dispose of the bomb, and then the officer makes a decision of what we're going to do. a muscle. And it felt, it was about the size of a grape, and I said, well, maybe I just pulled a muscle. So I waited until September 2014 to tell my primary care physician because I was worried about the lung. It's becoming bigger, and I thought I needed to have it checked out. I was sent over to the breast cancer center at the John B. Bertha Cancer Center at Walter Reed, and they did an ultrasound, and it was inconclusive. So they went ahead and did a biopsy on the 2nd of October. About the 14th of October, I was called into Dr. Craig Schreiber's office, and he gave me the news that I had stage 3 melanoma. When I heard that, I thought I was a dead man. I'd always heard that melanoma was a death sentence, and that, I was a goner. I was thinking about how my family was going to cope with this, and so on and so forth. Dr. Schreiber said that they would have to do surgery right away. I had to fill out paperwork, informed consent decrees, and given some permission to take my tissue that the doctor was going to remove from my side and put it in a biostorage area, hopefully for a pathologist to study it. On 20 October, my life changed forever. Dr. Schreiber resected one nerve node and 22 lymph nodes. The lymph nodes were sent to the laboratory, and on examination, only one lymph node had melanoma in it, and it had encapsulated, my immune system had encapsulated the melanoma. So I had, after I recovered from the surgery and my wound healed, I was sent over to Ellen Levy, she was a physical therapist because my arm had started recording the tissue. The tissue had started scarring, and my arm was starting to become hard to open. So she gave me some exercises to try to kind of balance the recording. In November, I went and saw Dr. Corey Carter at the hematology clinic. He gave me a choice I went there to discuss with him about chemotherapy or something else. Since my cancer was completely removed with the resection of my lymph node, I said I didn't need chemotherapy, but I did need to take an immunotherapy to ensure that my immune system had killed all the cancer in the rest of my body. So he gave me a choice between interferon, one injection daily for 12 months, or a new drug called immunotherapy. Try to say that 3 times. And said that I would have 4 some doses of that over a 12-week period. I chose to get me because I didn't want to spend a year injecting myself with interferon, and it sounded like it was a better drug for my situation. I then went down to see Dr. Stephen Lewis in radiology oncology. He told me that they would have to radiate my side to ensure that there was no residual cancer. And so we set up, he gave me a choice between the Australian method, which was smaller, less dose, but longer time, or the USA method, which is a stronger dose, but for a shorter time. I knew the Aussies and I knew they had a lot of skin cancer down there. I decided to take the Australian therapy. That lasted about 22 sessions. And then when I finished that, I started getting side effects. I had radiation burn on the side of my skin. I had my lung had been seared. My ribs were more brittle. And later on, I started getting lymphedema, which is these swelling of my right arm. After the radiation therapy, I was handed over to my chief Brad, and he gave me he ensured that I got the immunotherapy doses. I had 4 of them, and it lasted for about, I think it took about an hour and a half until I was each dose. When I finished the IMPI, then I was put on a monitored regimen, which I would go to the nuclear medicine clinic to have PET scans done, and then I would also go to the dermatology to check my skin. The problem with my melanoma is they didn't know where the primary site was. It ended up in my lymph node, but they still don't know where it actually came from originally.
Mark, I'm sorry to interrupt. This group is going to a break in just a second. Is it, if we certify that you did get through the IPI, you did get an autoimmune reaction where you had to battle through and you're on replacement Synthroid, is it possible to share what the big lessons are now? Thank you for what you shared. What is the things that you would want clinicians and researchers and other patients here in the audience to take home?
Well, first of all, it was very important that there was a team that was working on me. And also they were located in close proximity, basically on the same floor, the cancer center at Walter Reed. I also think that the patients need to get involved in their recovery. They need to be able to talk to their doctors and have a conversation about their cancer and how they're going to pursue the therapy. I also recommend that pathologists get more involved. I've yet to find out why my immune system encapsulated my melanoma and anything about the melanoma that I actually had. One other thing that I'd like people to know is that you get lymphedema. I'm told that your arm swells up, so they give you these sleeves, compression sleeves. This is the one that you wear at night. I call it the Iron Maiden because you put it on your arm. You can't bend your arm. It's supposed to keep the swelling from getting any worse. And then we have a daytime sleeve that we wear that looks like any athlete wears, like football players or basketball players. I found that those sleeves didn't do anything for me. Ellen Levy gave me some exercises to try to move the lymph fluid around by rubbing my chest and rubbing my side. That didn't do anything. I am a graduate of the Citizen Sciences Tools and Orientation course. It was given by Ananya and Steve and Andreas Gonzalez and Margie Fernak. This is my certificate to prove I was there. So using the knowledge that I gained by using the scientific method, my question was, could I improve establish more lymph nodes so that I can take care of my problem. My arm was swelling up, my hand swelled up, I couldn't even close my hand, and I wasn't comfortable with the sleeves. They would keep me awake at night, and nothing was happening. So my question was about how I can improve the lymph flow. I formed a theorem that I could do exercises that would, because after talking with Dr. Lewis and Ellen Levy, they said that it would also help moving your lymph fluid, because it's a passive system. There are no pumps pumping your lymph through your body. So I decided to test my theorem, and I came up with some exercises and stretches that I used. I started in about 2016, the summer of 2016, and I noticed after about 2 weeks that I was actually lowering the swelling in my arm and my hand, and it was actually measurable. The latest measurements that I reduced the swelling by 10%. Now, a lot of people were surprised about that, because normally a person with lymphedema will increase the swelling by about 1% a year. I myself said, I am not going to spend the rest of my life with sleeves and not being able to use my arm. I like to work out with the weights I run and all that. I wasn't going to be hampered by my arm. So I was doing the exercises, and I finally came in and showed Ellen Levy. She measured it. She has a machine to measure your arms, and it showed that I had actually decreased my swelling by at least 10%. And here are my arms. At one time, this is the one that was swollen, and this is my normal arm. And you can see, I couldn't see my knuckles or the bones in my hand or even my wrist. It was all swollen up to here. And through doing my exercises, I've been able to reduce it. I don't even wear the Iron Maiden, the night slave anymore.
Mark, if I can interrupt in closing here, and I'd like to see if we have questions for Mark. I don't know if you can tell that Ananya's turning herself inside out to hold this together. I don't know if we'll get him back. So I think if you have a couple questions, we could do it now. But briefly, I just want to say Mark is the type of patient that we are growing at Walter Reed that really takes ownership of the science. He's a collaborator in everything that happens, and he's actually showing us brand new ways to treat chronic problems like lymphedema that we didn't know. He's actually teaching our people some of the techniques he's done. He's actually weaning himself off of the synthetic steroids for his autoimmune disease. And these are remarkable patients. We have a lot of them now, and I'm hoping that as you start to develop your therapies and your research projects and your investments, that you'll have a patient like Mark in mind. Do we have any questions for Mark before we say goodbye to Mark? Mark, we're going to break. Thanks so much for your participation. It's great to see you. I look forward to seeing you in clinic soon.
Thanks so much, Mark.
We're going to break firm for lunch. We're going out the door at 12:30. Is that right?
Yeah.
Now at 12:15, we're going to do checkout. Okay? Remember that sheet of paper you have? We're going to flip it over and do it again.
The second half of it.
The second half, the bottom part, and we'll be doing it for 5 minutes. To make time go faster, Ananya and I have agreed to share the slides faster. I'll be doing this next section, and then right after checkout, she's going to be giving the conclusions. So are we ready to go?
Yep.
This is the big lift. This is you climbing up Mount Everest. Now you're getting ready to go, okay? You've got the background. You saw how we treat a patient like Dr. McCulley. You heard very intelligent patients like like Mark Wilhelmi. You've learned some cool tools that we hope you'll find useful. Now let's go. First slide. So we took these first questions that the research interns created, and it wound up by now, it's been going on, this process has been going on for a year and a half. We now have 15 questions, 5 in each of the areas. First question, what is the biological purpose of cancer? Why does nature allow cancer to happen at all? Next. With regard to this bad luck hypothesis, do you remember that all of this is mutation? Random mutation is causing all of this. How does this cause cancer cells to become so good at solving problems? How is that possible? Next question. How can the bad luck hypothesis explain how the cancer cells, not only how they start, but how do they solve problems when they get treatment? How do they always or tend to become resistant? Next question, why do normal cells switch to highly inefficient anaerobic metabolism, something called the Warburg effect that we're going to be going through, whenever they decide to become cancers? Why? And finally, if cancer is simply random, then why do we see almost exactly the same number of cases of cancer for every type? If it's random, how is that possible? Next slide. So now we're going to think about the evolutionary dynamics of cancer. Have you ever wondered what game cancer is playing? Why does nature allow this to go forward? Why don't normal cells just die whenever they get enough mutations? And here's something amazing. Because if you stop to think about how cancer could be working, if they are just going to take advantage of the host, don't they realize if they divide too quickly and kill the host, that they're killing themselves? So there ought to be a mechanism. Every other creature figures this out. Why not cancer cells? And apparently they have figured it out. You're going to hear some remarkable things here in this next few minutes of time. And it may not be totally relevant, but we think it may be critical to solving the cancer puzzle this year and to making America cancer-free. The game that cancer cells apparently want to do is they want to become immortal.
And we have references for those who want a paper that's been published on this. And we also have references at the end of the PowerPoint that link one will be sending to you later.
When you go to lunch, Ananya will have, she's copied all of the papers you're going to be hearing about, so you can just grab a paper on your way. Plus, every reference that you hear about will be at the end of the session. When you get the PowerPoint, you can just get the PDF file, okay? So you'll be able to go home and study this for yourself. Let's go. So this is the Tasmanian devil. This is a creature, a mammal in Tasmania, and it's very, very aggressive. And this is a Tasmanian devil with a tumor. Now, it turns out that this tumor is one of a series of breakthroughs that scientists have made in the last 5 years to understand what cancer appears to be trying to do. What is happening is these animals are very aggressive, they bite each other all the time, and whenever one of these normal ones bites an animal with cancer, he immediately, or almost, I can't remember exactly the incidence, but it's more than 90%, he's going to wind up with the same cancer. It will jump from one cancerous animal to the other. This is now considered to be a new species. These animals, this tumor has now learned to survive on its own, on the outside. And it's not unique. Next one. Oh, I'm sorry, go back, please. So it turns out that this has been going on for hundreds of millions of years. The clams and other shellfish do the same, so that they have transmissible tumors that they send as free life forms. You can classify a life form of one of two ways: the way it looks, the morphology, or its genetics. The Tasmanian tumor is considered now to be an asexual single-cell mammalian species. that is transmissible from one tumor to the other. The idea we want you to consider from this first slide is, what if the purpose of every tumor is to find a way to get to the outside and to become an independent species? It's certainly happening in many others, especially in the ocean and the invertebrates. And we would love to see some great research done in that area. Next slide. So, here's another problem that the students brought up at the very beginning. If you think of random mutations as adding entropy to a system, how could you get more intelligence out of the system? Adding randomness to a system only increases the entropy. And how could aggressive cancer be the only exception? If you consider, for example, a Rubik's Cube, it has 43 quintillion possible states. If you asked what's your chance of solving this with 1 billion random moves, it's almost zero. And so we want to rethink together. We'd like to get some scientists together to rethink how do purely random events, entropic events, other than protogene mutation into oncogene status, like the BRAF mutation, that causes a driver gene, a proliferation. The silencing of a tumor suppressor gene means that you have uncontrolled proliferation. That's not what we're talking about. We're talking about problem-solving intelligence. How does it figure out how to navigate all of the hurdles that it has to face, both inside the patient, from the patient, and also from the doctors? Here's an example. There's an article here. Ananya, could you please find the article? This is from Stanford. This is from the Stanford Medical School magazine. And please grab a copy of this if you're. It just came out last month. The basic message is this: that genes are followers, not leaders. What this means is simply this: that when you start to look at how the diversity of species develops in the natural world, it's probably not through random mutations. It's probably through organisms that take the genetic products that they have inside of them, and then like proteins, carbohydrates, fats, etc., and reconfigure them in ways that allow them to go into a high performance state. That's an epigenetic phenomenon. And then they start to modify the expression of genes with histones, etc., the way Ananya described. Over time, when that performance helps that species to do well in its environment, over time, then random genetic mutations will tend to sort it out. So this is a brand new tipping, hopefully you can see a brand new tipping point idea. Maybe this whole genetic mutation driving everything that happens is not true. Maybe it's epigenetic instead. So if you go back, please. Some of the areas that we really want to understand so much in cancer cells when they become resistant is, other than gene mutation, how do they reduce drug uptake? Is there other mechanisms that are non-genetic for amplifying drug targets? Mutating a drug target so it's not recognized seems to be very genetic. Failure to induce apoptosis may very well be epigenetic, as increased drug export. So these are just some of the examples of things that we have attributed to purely genetic events that may in fact be epigenetic. Next question. So here's a question. This is how normal cells in your body right now, as you're ready for lunch, this is how they metabolize sugar. If they are using an anaerobic metabolism, they are generating 2 ATPs for every glucose molecule. On the other hand, if they are in the presence of oxygen, then we can use the Krebs cycle, and they generate 38 ATPs. Why is it that when cancer cells are formed, when previously normal cells become cancer cells, they choose to only generate 2 ATPs for every glucose, regardless if there's oxygen or not? If you were trying to take over another country, why would you choose to use an energy system that's 1/16 as efficient? This is called the Warburg effect. It's been known for 50 years. We are using it when you do a PET scan. When you do a PET scan, we're looking for cells that hypermetabolize glucose. They're doing it for this reason. They need to take a pile of it up. And then, of course, when they do, it's a special FTG glucose that emits a positron, and that's what we look for. We think there's lots of other things we can do with this. What are the other ways? Okay, thank you, Ananya. Now, if cancer is caused primarily by bad luck mutations, then why are the incidence rates of cancer the same? This is breast cancer, for example. For the last, say, 15 years or so, it's a flat rate. Almost all of the cancers are that way. We almost always have the same number of CML cases or melanoma. There's a few cancers that increase, like, for example, using tanning beds for melanoma, but we're really not sure for the reason of that. As I mentioned, it might be mutations of melanocytes, but it may actually be damage to the immune system that's in the environment. And we would like to get a group of researchers that could explain how does this happen by purely random events. When I talk to people in the SEER data, etc., they really don't know. Okay, we're now at question cluster number 2. Can I ask what time it is now, guys? Because remember, we're going to stop at 12.
It's 11:55.
Okay,
We're stopping at 12:15.
Yeah, so we're on track. Okay. So now we're in the second cluster of questions. Are immune system cells intelligent? And what wonderful feedback we've already got. What do you mean intelligence? How do you measure that? Is that really something that scientists can work with? Well, and when you come to be a patient with me, what I'm trying to figure out is how intelligent is your immune system to recognize cancer, and what would be the best strategies that I could use or that we could develop together that could help your immune system upgrade its ability to recognize and fight cancer. That's a new motion that I consider to be highly intelligent. So, can you go back, and now you were on 6. So the first question is, what's the role of first responder cells? Go to as many cancer meetings as you want. AACR is just happening today, as you know, many of you chose not to go there. And all you're going to hear about is about CD8 positive killer T lymphocytes. And we got a question during the break about Tregs. What's the role of Tregs in tumor specimens to keep the patient immunosuppressed? And how do we get around that? It's all second responder cells. And yet, not only in the Macaulay specimen, but in other specimens that we biopsy, why do we keep seeing mainly first responder cells? What is PD-1 and anti-PD-1 doing to them? Why are they there? There's no research being done in this at all that we can find. And we think that this is a fruitful area for hopefully people like you to consider. Okay? The next one is, since pre-vertebrate animals don't have T or B lymphocytes, but they do get cancer, how could we use pre-vertebrate research models? The next one is, why do normal immune system cells also do the Warburg effect? Next question. And how can we require pathologists or inspire pathologists to start to comment, give us information about the aggressiveness of tumor and about the immune system cells? This is something that Mark Wilhelmi was just mentioning. And finally, how can we transform the attacking terrorist cells with cell poisons, phase 1, phase 2, phase 3 clinical trials design that we've had for, gosh, it's been 60 years, with something that's actually specific for immune system stimulation. It has to be a different model. Next slide. So, once again, when you start to think about immunotherapy of cancer, get ready to hear about this. And get ready to hear about the transformation of this to this. And to a lesser degree, get ready to hear about T lymphocytes. But what about all of this? What about how does this all fit in? And why is it that we mainly saw these kinds of cells in these tumor specimens of patients, not just Dr. McCullough, but other patients? No one's doing research on this to the best of our knowledge. And more importantly, perhaps most importantly, as we're going to go back, when you start to think about a cancer-free America, how would we start to think about the immune system so that normal people's immune system could stay elevated intelligent, fighting cancer, it's probably not T lymphocytes that are doing that. If you have 5 or 6 or 20 cells today that are becoming cancerous, it's not very likely that you're developing a T lymphocyte response to it. It's probably these macrophages. They are the first responders. They probably know exactly what's going on, and they know how to eliminate it. We would love to see research in this area. Yes. So here's another interesting area that came up in our questioning. I had no idea about this. When you start to think about multicellular organisms, more than 95% of the multicellular organisms do not have a backbone. They're pre-vertebrate. Maybe more than that. And only here do we start to see animals with backbones and mammals and humans. When you are below this line, when you're an invertebrate, you do not have any T and B lymphocytes, 0. It's only one first responder cell. It has different names, immunocyte, hematocyte, but it's basically the invertebrate equivalent to our mononuclear phagocytes. We know that these animals get cancer. We know they get melanoma, for example. How are these animals with their immune systems combating cancer with their immune system? To the best of my knowledge, there's no research being done in this area at all. And it seems these would be very inexpensive models to start to explore how the immune system could be, at the first responder level, could be working in cancer. And the reason this would be super important is because, again, if you start to believe that everybody has cancer all the time, then the assumption would be it's not second responder cells that are eliminating it, it's first responder cells. Just like with a fire, it's so rare to see a 3 or 4 line fire because it's usually first responders can do a good job. This is the same slide that you saw with the cancer cells. This fact that normal cells, including normal immune cells, under normal aerobic conditions will make 38 ATPs per glucose. When they get turned on with anti-PD-1 or anti-CTLA-4 and they start to go into hyper-attack mode, they do exactly the same thing. They reduce to 2 of ATP. Now, why would that be possible? Why, if they're going into attack mode, why would they voluntarily choose less energy supply? But that's become a huge clinical problem because when we see nodes that light up in our patients, we don't know if this is advancing cancer or immune system. We need research on this to distinguish this. For example, is there any difference between the Warburg effect in cancer cells versus immune system cells? We could help so many patients to avoid biopsies, etc., because this is becoming a huge problem if we could start to have researchers that would study this. Can I see a show of hands of anybody here who studies the Warburg effect? Yes, but this is great. Let's talk, because this is an area of huge clinical importance for us right now. Next slide. When you get a biopsy, when you or a loved one goes in and gets a biopsy, they're going to find out so much about the cancer. Everybody wants to know, do I have cancer, yes or no? Next. And then, for example, here's the cancer cell. Right here, you've learned to recognize it from the Macaulay slide. But then what about these cells? This is a polymorphonucleoleucocyte, etc. You're not going to get any information. You're paying for the pathologist to look at it, but you're not going to get any information about what does the surrounding cellular type look like. This is something that we desperately need to get. The pathologists are very bright people. They have their standards, and this is not one of them. The field has changed very fast, and the pathology has not kept up with it. If any of you are going to get a biopsy soon, I would make sure the pathologist gives you as much details as possible about what does your white cells look like. Who's there now? And then if you're able to get a treatment, you may consent to do multiple biopsies to see how these guys are responding. Because remember, when you get an immunotherapy like anti-PD-1 or anti-CTLA-4, we're not attacking the tumor cells at all. We're empowering or upregulating the intelligence of these cells to recognize and eliminate. And there's so many first responder cells and other cells that are in specimens, but no one's systematically gone through it to understand it. One last thing. If you think about your immune system as being unique, if you were to come into my office, I would tell you that your immune system appears to be an extension of your nervous system. Every one of your nervous systems is unique. It has different genetics, different cellular structure, and different experiences that make the neuroplasticity happen. None of our two immune systems are the same. And yet what they do is your immune system recognizes entropy forces, it recognizes the forces of health and meaning, and then it helps you to make the choices towards health and meaning. That's how you drive here, and that's how you're going to get home successfully. Your immune system is doing exactly the same thing, except it's doing it for 100 trillion cells, 24/7. And everyone's immune system is unique. No two people have the same zip code. We all have different zip codes that our immune system is patrolling. More than that, as we'll be building the case later on, if you can see how many players there are in the immune system, and then there are now hundreds of signals. We're going to be hearing about one of them after lunch, IL-10. But there's hundreds of them. It's like an orchestra that's going on there. It's got to be timing and intensity, timing and intensity. How do we start to learn more about, from each patient's experiment, how that happens? The day of these huge clinical trials where we have 5,000 breast cancer patients all getting the same chemotherapy, and then we're looking for a 20% reduction. Do you see that that's changing? That won't work in this. Every person's immune system is unique. Every person's reason that their immune system is allowing cancer to happen is certainly unique. And then every person's response to the treatment, including autoimmune effects, is unique. We need a research model that addresses this. And I'm so glad we have young people here that can think about it. Okay. Here's one of them. Clinical trial design. Our clinical trial design, number one, Ananya, what we're looking for today is the dose of the drug that we can give that won't kill the patient. But that's because we've been held hostage for the last 70 years to poisons that we give that kills the cancer. We're like trying to carpet bomb ISIS. That's how we think. So then what's the maximum dose of the poison we can give and not kill the patient, right? Next, then what we're looking for is to give that maximum dose to as many people as possible to see how effective it is. And then finally, we compare that, the new treatment at the maximum dose to the established treatment to get final FDA approval. This is what we need. We need to find the optimal dose that boosts the cancer-fighting intelligence of the immune system. That's not going to be the maximum dose that almost kills you. And no wonder we get so many autoimmune side effects from these agents, because we know we're giving way, way too much of this. Maybe in the order of thousands or even millions of times more than we need to give. We need to look for the effectiveness of the optimal dose in each patient, and that won't be the same for a big group. And then we need to compare it to established treatment. This is something we can do right now. We know we're giving the wrong doses of these agents. We're just giving astronomical doses. Imagine your favorite symphony, whatever you love, the music you love to live. And then imagine me with the tuba playing it as loud as I can. Ba, 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 ba. That's what we're doing. It doesn't make sense. And we could, as a group, we could expand this type of conversation so that we could sit down with FDA. There are wonderful people. They're not stupid. They just need to get critical scientific feedback. And it's time for this issue to come forward. It won't cost one penny to start a new trial design mechanism. And I would urge this for any of you who are starting companies in this area that will focus more on optimal dose finding, not maximum tolerated dose. That's the last century. Okay? How much time do we have now, Ananya?
It's 12:09.
12:09. All right, we're going to
Yeah,
blast
5
through.
minutes.
Okay, I'm going to go climbing up Mount Everest. Okay. if I had to sparkle one thing that would change everything for all of us immediately, is to prove this. How does everybody have cancer all the time? Number two, how can we prove we have something called spy versus spy going all the time? Number three, what is the abscopal effect? I may not get to that. How can we diagnose the unique ways that each specific cancer patient's immune system fails? And finally, how can we ensure that new trials for cancer, for treatment of cancer, also shed light on how the immune system protects normal individuals? We can do that at the same time. This is called the Philly Lander, and this machine is sitting on a comet 200 million miles out in space. I think it was landed there successfully about 4 years ago or 3 years ago. The comet is 6 miles wide, and this thing is about the size of a Coke machine. You guys know about this? Yeah? And this machine chased the comet for 4 billion miles to land successfully and transmit data. You're shaking your head, okay? If we can get that level of sophistication, the people who did this are not gods, they're just fellow scientists. Why can't we start to develop nanotechnology that can go into the patient's cell and prove that everybody has cancer all the time? I am volunteering to be the first person to receive the injection of whatever it is. We need a critical mass of people who want this experiment to take place. It will revolutionize medicine as we know it. Next slide. This is from a cartoon, an American cartoon series, Spy versus Spy. And this is kind of how I'm seeing cancer every time I see it. I see my immune system is unique, but these newly emerging cancer cells are all trying to get to the outside. They're trying to do this Tasmanian devil thing. You can actually see this in Dr. McCulley's case. You remember that tumor going out, making a break to become immortal. How do I give my immune system the upper hand? We don't have research that really sees it this way, and the reason for it is because these cells are not seen as intelligent problem solving. They're seen as just cells that do what they do because of random mutations, sometimes random hypermutations. Let's pass over the abscopal effect. Now, so this is the penultimate question. When you start to look at a diagram of how the immune system looks, it starts to get really crazy. Here's a couple of first responder cells, but you like dendritic, but you know there's many, 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 many more doing anti-cancer things. Here's some of the second responder cells, and this is how they're processing fragments of tumor. But then when you start to look inside, this, honestly, anybody who's an expert in this will tell you, this is 1,000th of what we now know about all the signals that go back and forth in receptors. It's just crazy. For us to believe that every cancer patient has exactly the same defect here, when we give an anti-PD-1 or anti-CTLA-4, would be like every time any one of us bring our cars into the shop, it always has a carburetor problem. Really? It couldn't have a transmission problem or a steering or a brake? No. It's time to start to want to understand what are the specific, unique ways that we can start to measure and do a profile of cancer patients to see this is what broke down. And that research is not ongoing right now. Last slide. So here's another idea. When you start then to look at the whole process of normal cells becoming aggressive cancer cells, and a series of mutations that certainly do happen, but how much of that does it explain what they do? And certainly at some point, they can really become quite dangerous in their problem-solving ability. But we now know there's a mirror world, this for sure, that immune system can recognize and eliminate at early stages, and can also recognize and eliminate late stages. Next clip, please. What we'd like to, go back please, what we'd like to do is to please consider designing treatment trials, like the McCulley case that you saw, that help each patient get the treatment that they need with their immune system, and at the same time design them so they could tell us more about the mechanisms that's happening in all of us all the time. This is possible to do. Remember, Jonas Salk says the answer pre-exists, it only requires the right question. We need a much more intelligent way of developing these drugs, approving these drugs, and then as we design the trials, to make sure that from each unique patient, we help that patient get the victory they need in a way that tells us more about what's happening in each of us all the time. And with that, I think we're ready for checkout, is that right?
Yep.
Do all of you guys still have your sheets of paper? Does anybody need a pen? Because what we like to do is another 5 minutes to put your heart on this piece of paper as to what you've learned about cancer. It may just be new questions you have about cancer. At the end of the day, here's what I'm claiming for you guys. There's only one scientist you'll ever believe. That's you. And this is your chance to see how much you learned. Is it okay if we do 5 minutes really quick? Ready? Go.